Assalamu alaikum dear students, I am Muhammad Rauf, lecturer Department of Zoology Cust. Welcome to the course Principles in Animal Life 2. This is lecture number 20. The topic is Chromosomal mutations. Discussing agenda for the topic is alteration of chromosome structure, aneuploidy of sex chromosomes, Down syndrome, Trisomy 21. Lecture outcome. After watching and listening this lecture, students will know about various types of mutations in chromosomes and their consequences. This is the second part of the topic, and we will recall what we studied in the first part. So chromosomal mutations are changes in the number of chromosomes or changes in the structure of chromosomes. Change in number of chromosomes is of two types: aberrant euploidy, when there are multiples of the basic set of chromosomes, and aneuploidy, where one particular chromosome is either in excess or deficient, means 2n-1 condition, which is called monosomy, and 2n+1 condition, which is called trisomy. Now the 2n-1 condition, the chromosome which is missing can either be a sex chromosome or an autosome. Similarly here, 2n+1 condition, the chromosome which is in excess can either be an autosome or a sex chromosome. Example of that particular condition is Turner syndrome. Example of that condition is Down syndrome. Example of that condition is Klinefelter syndrome. Here in that particular video, we will discuss the changes in the structure of chromosomes, which are of four types: deletion, duplication, inversion, and translocation. So let's start. Changes in structure of chromosomes or alteration of chromosome structure. Errors in meiosis or damaging agents such as radiations can cause breakage of a chromosome. The chromosomes are usually fragile at the ends, so there are chances of breakage at the ends, and when a segment is detached from one chromosome, then this segment can attach to another chromosome because of having sticky ends. So deletion is quite simple, where a segment of a particular chromosome is detached and missing. Now that detached segment, if has a centromere, then it will be part of the gamete when that particular cell undergo meiosis, because it has centromere and it has the site for the attachment of spindle fibers. But if it lacks a centromere, then such a segment is called acentric. Acentric means without a centromere, and because this segment does not has any site for the attachment of spindle fibers, so it will be lost from the organizing nuclei or the newly formed cells. Sometimes the detached segment can also attach with a sister chromatid of the same chromosome, and in such case duplication will occur because the sister chromatid already has that particular segment and a segment which was detached from the other sister chromatid attached to it, so there will be certain genes which will be duplicated here, and this phenomena is called duplication. Duplication can also arises on its own where a particular segment of a chromosome is duplicated, and recall from the lecture on gene mutations, we said that gene mutations are responsible for the evolution of new alleles, and because here are four types of gene mutations, four types of changes in the chromosomes structure. So here this particular segment is duplicated means the genes which are located in that segment are also duplicated. Now what will happen? The genes located in the original position will perform their function normally, and the genes which were duplicated, now they are available for the natural selection, and they may evolve into performing some other related functions, and duplication is responsible for the evolution of gene families. Gene families are such group of genes which performs related function, nearly related functions, like the in hemoglobin we have alpha and beta chains. So these two chains are produced by genes which are closely related with each other. So remember that duplication events are responsible for the evolution of gene families. Here we discuss another type of chromosomal mutation, change in chromosome structure, inversion. Now in the previous slide we studied duplication and deletion, where there is a net loss or gain of chromosome segments. In inversion there is no net loss or gain of chromosome segment, only a rearrangement. So what happens in inversion? This is a normal chromosome, and that particular segment is broken from both sides. It is rotated at 180 degrees and reattached with the same chromosome. So what will be the effect? Only the order of the genes will be changed, whereas there will be no net loss or gain of genes. Translocation is another type of chromosomal mutations. Now recall the process of crossing over which occurs between homologous chromosomes. So in translocation the segments are exchanged between non-homologous chromosomes. So if segments are exchanged between non-homologous chromosomes, then it is translocation, and if segments are exchanged between homologous chromosomes, then it's a normal vital process responsible for genetic variability, and it becomes crossing over. Now what are the different consequences of chromosomal mutations in humans? Alteration of chromosome number and structure are associated with serious human disorders. As described earlier, non-disjunction in meiosis results in aneuploidy, means either a chromosome copy is missing or in excess. Most of the chromosomal alterations are so disastrous to development of the embryo that the affected embryos are spontaneously aborted long before birth, means those chromosomal mutations are lethal at times, and the delicate embryo cannot afford to carry such a mutation for too long and dies away. However, some type of aneuploidy appear to upset the genetic balance less than others. Means there are few cases where although there is a genetic imbalance, there is a loss or gain of a chromosome, but still the individual somehow makes it till birth and beyond. These individuals have a set of traits or symptoms which are called syndrome. Syndrome is a set of symptoms and characteristics of each type of aneuploidy, means whether a chromosome is missing or extra, or if the chromosome is missing, whether it was a sex chromosome or an autosome, or the chromosome which was an extra copy, whether it's a sex chromosome or an autosome. So all those conditions comes under aneuploidy, but they have their characteristic symptoms. Genetic disorders caused by aneuploidy can be diagnosed before birth by genetic testing of the fetus. Suppose if there are certain vulnerable factors, means the age of both the parents is very high, or the parents were exposed to some kind of mutations or chemicals, then in countries there are certain tests which helps the doctors to evaluate the genetic complement of the developing embryo and the health of the individual chromosomes. So this process is called genetic testing, and it is specifically called prenatal testing because it takes place before birth. Some of the cells of the developing embryo or from the amniotic fluid are taken and they are analyzed for the presence of any abnormalities, and just in case if there are abnormalities, then the parents of the individual are informed and they can take decision about whether to carry on with the pregnancy or to abort the individual. So the tests which are commonly in practice are chorionic villus sampling, amniocentesis, and pre-implantation genetic diagnosis. Amniocentesis and chorionic villus sampling is done at some stage after pregnancy is induced, and the most latest type of test which is in practice is pre-implantation genetic diagnosis, where the sperm and the egg are taken from both parents. They are fertilized in vitro outside in a test tube, and after fertilization the proper conditions are provided and the zygote starts developing and dividing, and after some time some cells are taken from it and analyzed. So if the cells carry a genetic disorder, then the embryo is discarded, or if the development is normal, then it is placed in the uterus of the female from whom the egg was taken, or into a surrogate mother that is in foreign countries. But pre-implantation genetic diagnosis is controversial because people use it to find out the gender of their offspring. Now, Down syndrome, trisomy 21, it is the most widely studied syndrome or chromosomal abnormality, and as I already mentioned, that aneuploidy in autosomes is usually lethal because autosomes carry imported genes which are responsible for maintaining the normal life of the individual, but it is a unique case where aneuploidy occurs in autosomes and still the individual makes it till 15 years of age, and 8% of the individual makes it till 40 years of age. So trisomy 21 means there are 3 copies of chromosome number 21 in those individuals instead of the normal 2. Here you can see this is chromosome number 21 and it is present in 3 copies instead of the normal 2, and on the right side you can see a child affected with this condition. So when 3 copies of chromosome 21 are present, it means it's a 2n+1 condition, and the chromosome complement of that individual will be 47, and it is called trisomy. Don't get yourselves confused in trisomy and monosomy. It's pretty much simple. Monosomy is when one chromosome is missing. Trisomy means when one chromosome is in excess. So just in the previous slide we studied that each type of aneuploidy has their own characteristic symptoms. So what are the symptoms associated with Down syndrome? They are characteristic facial features. Short stature means these people have lesser height, and they have cardiac defects, but they are correctable, means they can be treated, and they have developmental delays, means the puberty might come later than in normal individuals. And individuals suffering from Down syndrome are more vulnerable to acquiring leukemia and neurodegenerative disorder called Alzheimer's disease, but they have a lower rate of hypertension and atherosclerosis, which is hardening of the arteries, stroke, and many types of solid tumors. And let me share a general knowledge here is that the World Down Syndrome Day is celebrated on the 21st of March every year. Why specifically 21st of March? Because March is the 3rd month of the year, and 21st signifies the fact that in trisomy 21 3 copies of chromosome 21 are present. So 21 is the date and March is the 3rd month of the year. Almost all males and about half of females with Down syndrome are sexually underdeveloped and sterile. The frequency of Down syndrome increases with the age of the mother, while the disorder occurs in just 0.04% of children born to women under age 30. The risk climbs to 0.92% for mothers at age 40 and is even higher for older mothers. The correlation of Down syndrome with maternal age has not yet been explained. So this is basically a statement from the Campbell biology book, but in other books they have suggested a reason that why the instances of Down syndrome in the children's increases when the age of the mother increases. It is because of the process of gametogenesis. Gametogenesis, that is oogenesis, starts in the human female even before the birth of the female, when the female is itself developing in her mother's womb. But this process is arrested in meiosis 1, prophase 1 stage, and when the female is born, the female reaches puberty, the hormonal levels reaches to their normal, which stimulates the development of those eggs, and that process which stopped before birth is started once more at puberty, but is arrested again in the meiosis 2 stage, and will only be completed after fertilization, after the egg is fertilized by the sperm. So let's imagine a woman is 40 years old, so means the process which started before her birth was interrupted and arrested for 40 years. So there will be more instances of non-disjunction as compared to a woman having 20 years or 25 or 30 years age. The medical experts recommend prenatal screening for trisomies in the embryos to be offered to all pregnant women due to its low risk and useful results. Means prenatal testing we already discussed, so it is recommended in cases when there are vulnerabilities, when there are chances of abnormalities, and these tests are simple, they are risk free, and they are beneficial. Now, the previous example, Down syndrome, was aneuploidy in autosomes. Here we study aneuploidy of sex chromosomes. Aneuploid condition involving sex chromosomes appear to upset the genetic balance less than those involving autosomes. We have already discussed that statement. This may be because the Y chromosome carries relatively few genes. Also, extra copies of the X chromosome simply become inactivated as bar bodies in somatic cells. An extra X chromosome in a male producing an XXY genotype occurs approximately once in every 500 to 1000 live male births. Means an individual born with that particular chromosome, a frequency is 1 out of 500 or 1000 male births. So Klinefelter syndrome is aneuploidy in sex chromosomes, and it is a trisomy where one of the chromosomes is in excess. So the male individuals affected with this condition have male sex organs, but the testes are abnormally small and the man is sterile. Female can also be affected with Klinefelter syndrome, but the females are lucky in a way that if the X chromosomes are above normal, then they have a normal process which is called X chromosome inactivation. So the X's copies of the X chromosome are inactivated, but those females do exhibit some symptoms because the X chromosome inactivation process is not 100% effective, and some of the genes skip the process of inactivation. So what will be the effects in the females? There will be enlargement in the mammary glands and other female body characteristics are common. Affected individuals may have subnormal intelligence. The instances of acquiring this condition is 1 out of every 1000 males that will be born with an extra Y chromosome. The male undergoes normal sexual development and do not exhibit any well-defined syndrome. Females with trisomy, which occurs once in approximately 1000 live female births, are healthy and have no unusual physical features other than being slightly taller than average. Why they don't have other unusual physical symptoms? I have answered that question. XXX females are at risk for learning disabilities but are fertile. Monosomy X. Now this is monosomy. Remember, Klinefelter syndrome was a trisomy in sex chromosome where one copy was in X's. Monosomy is a condition when one copy of the sex chromosome is missing. It is 2n-1 condition, and the example is Turner syndrome. Now why they have designated these syndromes as Down syndrome, Turner syndrome and Klinefelter syndrome? It is because these names signify the fact that these conditions were for the first time reported by those scientists. So the Down, Turner and Klinefelter are the names of the scientists who for the first time reported those conditions. Although these X0 individuals are phenotypically female, they are sterile because of their sex organs do not mature. Now, monosomy is a condition when one of the X chromosomes, one of the sex chromosomes is missing. So those females will have a chromosome complement of 2n-1 or 45, and they will have only one copy of the sex chromosome. The other copy is missing and represented as 0 or 0. When provided with estrogen replacement therapy, girls with Turner syndrome do develop secondary sex characteristics. Most have normal intelligence. So those individuals lacks estrogen, which is responsible for the development of the sexual characteristics, and when estrogen is supplied to them, then the development of secondary sex characters occurs and they have normal intelligence. This is the end of the lecture. Thank you very much. Allah Hafiz.